Welcome to ISL Enabled Virtual Lab of CSIR.
Welcome back. And now we have with us Dr. Karishma Kaushik. She's a scientist who works on innovative approaches like biofilms to understand infectious diseases. Her lab specifically focuses on understanding the role that microbes and biofilms play in diseases which are relevant to humans. So, Utsuka, why don't you go ahead and ask your question to Dr. Kaushik?
So in our previous discussion, we learnt about biofilms, and we also learned that biofilms have so many different kinds of microbes living in them. So our question to you is, how do you study these microbes in your lab?
Great question, Utsuka. There are different ways of doing this. One thing is the oral cavity has a huge diversity. So we can't replicate all of this diversity in the lab. So one approach we do is to break it down and take few relevant microbes based on previous research and previous studies. The simplest model to study them in the lab is to do them on the bench, the laboratory bench. And so for this, we use something called in vitro models. So we grow these microbes on plastic dishes, surfaces. A nice way to make it more relevant to humans is to add factors that the microbes would likely encounter in the human body. So what do you think could be something a microbe encounters in the mouth? Sugar. We love fruits. We love chocolate. Coffee. What else can we think of? So we can actually add these factors in the lab when we are growing the microbes and study how they behave in them when they form biofilm. They're simple in the sense that they don't recap all the factors in the oral cavity, but they are interesting enough and they provide some good insights and in an accessible and affordable format. The next step is to make larger and complex models where you can add multiple different microbes, multiple different factors, study the effects of these factors on the microbes or microbial growth, biofilm composition over time. You can also look at it under the microscope. And then we have advanced models where we can use animals to study biofilm infections. Common animals used are mice, rodents. Of course, animal models do present a range of ethical concerns, and therefore we should be very judicious and careful in thinking of the kind of experiments we need to do with them.
How do you get the microbes from the oral cavity?
So usually, Jigyasa, we start with the lab grade microbes. We can use clinical samples, in which case we'll access them through mouth washings or scrapings or brushings, or in certain cases, even tissue biopsies. But then you're only sampling one person. So it's not representative of the whole population because I may eat something that you don't eat.
So you just now mentioned clinical samples, right? We also know that you are a clinician come researcher. So how does your unique background help you do your research with biofilms?
Honestly, when it comes to my professional trajectory, I think I don't know how to describe myself, but yes, by training and education, I'm a medical doctor. And now I work predominantly in the role of running a research group. It's been an exciting journey, right? Lots of wonderful things happened. Like I discovered my love for research during this time. I also returned to India, which was a very deep commitment and motivation I had to come back and contribute to our science ecosystem. How my research differs from possibly many others who are purely scientists in their training is that I always like to see the big picture in mind. I always like to see how this can possibly impact healthcare practices, technologies, patient welfare. What I feel is I start with the other end. I start with what is the patient problem and then try to get that in the lab.